Need milligrams of complex eukaryotic protein fast? Welcome to the insect cell powerhouse that turned a caterpillar virus into modern biotechnology's Swiss Army knife. From 1947, polyhedra in silkworms, to 1983, when Smith and Summers swapped polyhedrin for human bee interferon, BEVS was born, DNA viruses that infect only insects, zero risk to mammals. We delete the occlusion gene and hijack the ultra-strong polyhedrin promoter to overexpress our target protein. Clone gene into an expression vector like P-Fastback, recombine to make a BACMID, transfect SF9 cells to recover P0 virus. Amplify into a high-titer stock for infect production cultures and harvest at 48 to 72 hours. Then clarify and purify to get your ready protein. Total need: 14 days or less from DNA to milligrams of material in many cases. Choosing the right insect cell line determines yield, secretion, and glycosylation. How to choose your cell factory. SF9 and SF21. Robust, fast, great for intracellular and membrane proteins. High 5 cells, secretion champions, often 2 to 10 times the titer for secreted targets. Glyco-engineered insect lines give more human-like N-glycans for therapeutic projects. E. coli, cheap and fast, but often inclusion bodies and no eukaryotic PTMs. CHO, perfect glycans for therapeutics but long timelines and higher cost. Choose BEVS, 10 to 14 days, 50 to 1,000 milligrams per liter, eukaryotic PTMs, the sweet spot for 80% of projects. Let's see some case studies such as COVID-19 VLPs, scaled to 1 billion doses in 2 kiloliter bioreactors, HPV vaccine manufacturing, 40 to 60 milligrams per liter of L1 VLPs, ready for a lung absorption, ion channel tetramers, 200 micrograms per liter for cryo-EM structural work in about 3 weeks. Next part, optimization hacks and recent advances that speed development and stabilize yields. Tips: codon optimize your coding sequence for Spodoptera codon usage. Use a his tag plus TEV cleavage site for single-step capture and native removal. Run a multiplicity of infection matrix from 0.1 to 3 to find the functional expression peak. Harvest when culture capacitance drops by about 15%, an easy functional trigger. Arginine and SEC buffer is a practical trick to reduce aggregation during polishing. Let's see some new tools for BEVS. CRISPR knockouts to reduce fucosylation for improved therapeutic glycoforms. Flashback next-gen streamlines vector construction, no plaque picking required. High-throughput 96-well automation can scout micrograms of expression in about 48 hours. Perfusion processes now reach over 40 million cells per milliliter for intensive production runs. Thanks for watching. Find more information of BEVS and protein expression service on our website.